D.9 is looking at how do we actually design the drugs that we use and administer in pharmaceuticals. When we're going through and designing a new drug, we need to look at how molecular biology works. And so this is where a lot of overlap occurs with the human body because it is the human body we are working with. So we know that by interacting with and binding to key biological molecules like an enzyme, a receptor on a cell membrane, or DNA, a drug can prevent or inhibit its normal biological activity. So it can interrupt the development of the disease. So specifically, if we're looking at antibiotics, we are looking at what can we inhibit on the bacterium themselves to prevent the bacteria from growing and spreading, so on and so forth. So what occurs when we are going through and actually designing and looking at new drugs? And because we briefly started looking at the process in D1 of how new drugs get through the FDA and onto the market. What they're doing now with the technology we have is creating compound libraries. This is where we're going to identify lead compounds for the target molecule. So we discussed this back in D.1, where we have an idea of what kind of molecule will target, or what kind of plant or biological secretion could target a disease, whatever it happens to be, and then we try to figure out what molecule within that solution is, or substance, whatever it is we're looking at, is actually targeting the disease itself. So we've moved to what's called a rational drug design. And it's very different from the time when pharmaceutical companies worked mostly on trial and error basis, where we'd start with that natural remedy and try to improve on nature by creating the synthetic version, but we really didn't know how, so it was that trial and error. But by using the technology we have today and our understanding of biological processes, we've really made an efficient process in identifying what molecules are what. So we've targeted synthesis and testing of new molecules based on this current knowledge. What these libraries do is they are taking all this information that we learn about all these different compounds we are testing. We are cataloging all the tests for these compounds, and we're going to store them for future purposes because maybe this compound that they're looking at is not targeted for the disease they're trying to work with, but it may work for the next disease. So they don't want to lose that information. So we create a library of knowledge of the compounds that are being used for the pharmaceutical purposes. They're going to create all the biologically relevant information through the chemical screening process used. And we're going to talk about all these different processes in which they use. We have that need for efficiency in drug designing because our knowledge of diseases and understanding of diseases is becoming quicker and faster and more efficient. We want to make sure that the drugs we are putting out is faster than has been in previous years, so we can start treating patients quicker. The different type of processes we're going to talk about for creating these libraries is combinatorial synthesis, parallel synthesis, and high throughput screening. Looking at combinatorial synthesis first. This is a process of synthesizing groups of compounds simultaneously. So combinatorial libraries is what we're creating. So they can look at anywhere from 10,000 to 500,000 compounds. Essentially what they're doing is running a lot of mini reactions all at one time because we have the technology in order to do this. And we can be collecting data through the computers and through the analyses that are going to be run. So we can create a pool of information. So we're going to have these mini small-scale synthesis reactions. And there's going to be a variety of starting materials and reagents depending on what it is we're looking for. We're going to screen those products. We're going to look for the desired activity, and that's going to identify that lead compound for us. So traditionally, we would look at A plus B would give us compound AB. Now, because we have the technology to run these small-scale synthesis reactions, we could have A1 reacting with B1, B2, B3, so on and so forth. And then A2 reacting with B1, B2, B3, and we have this almost limitless possibilities, creating multiple different types of compounds, giving us a lot of different information. What is really interesting about this is it mimics natural processes. So if we look at natural selection and how species survive and what they go through during that natural selection process, a lot of it is random mutation. So your DNA is just randomly mutating and it happens to be beneficial, so you're going to survive, or it happens to not be beneficial and you're going to be more likely to die, vice versa. But we are essentially doing the same thing where we are randomly mixing multiple different types of elements or compounds together to create new products. And at some point, we are going to hit the target product that we are looking at. There are a couple different processes and how they do this. The first one we're going to talk about is called solid phase chemistry. This is specifically looking at what are called peptide combinations, and peptides are proteins. And so these are making very specific types of drugs. Not all drugs are protein based. It all depends on what it is we're looking for. So to kind of give you an idea of what this is the solid phase uses a resin in which the amino acids are going to be attracted to in order to start to build upon each other. So to kind of give you an idea of what this looks like is we have our resin bead. On that resin bead, what we put into the resin bead solution that we have is a peptide. And so we have that peptide, and we're like, okay, so that gives us one amino acid. We're like, that's not the amino acid we're looking for, so we're going to attach another one, and we're going to attach another one, so on and so forth until we get the combinations in which we need. But the beauty about this resin bead is we can be putting on multiple of the same strains, allowing us to create a lot of one type of compound at one time, knowing exactly what the combination is, because we're only adding one amino acid at the time. And then when we are all done, we are going to go through and cleave it. So one of the processes is called mix and split. And so this is the most common of the solid phase chemistry that we use. So what we do is the components are linked to the solid support resin bead, which is what I just discussed. They're going to mix it and then split it. And so now we have a different combination. So depending on how it gets split, we're going to have different combinations. Then each portion is going to be reacted with different building blocks. And so we're creating multiple different types of peptide strains, so different colors like I had shown you. And then you're going to mix and split and repeat, so on and so forth. So if you think about, we have 20 amino acids and we can have a combination of those. That is 3.2 million different combinations possible during this mix and split and the creations, and depending on how big you want to make the chains, so on and so forth. So there's a lot of different situations in which we can use, and all that information is going to be put into the libraries. However, we are actually starting to move towards more of a solution phase combinatorial chemistry, replacing solid phase, because it allows us to synthesize smaller molecules, and because it's in a solution, they're more likely to be able to be put into a pill form. The problem with the solid state in creating specifically only peptide style drugs is they cannot be taken orally, because pepsin is in the stomach and pepsin breaks down to peptides. And so you're not going to get the desired effect of a peptide style drug. That needs to be more of an intravenous. So for ease of the type of drugs available, the solution phase is going to be the non-peptide molecules, so that we can take it via pill form, because lipase, which breaks down more of the lipid style drugs, is going to happen within the intestines where we're going to absorb it. so on and so forth. So depending on the type of drug we're making, we have 2 different versions. Solution phase is very similar to where we can mix and split and make a variety of different compounds, but it's just the different style of drug we want to make. The next series is parallel synthesis. Parallel synthesis is going to produce a single product, unlike the mixture created in combinatorial synthesis, where we're making anywhere from 10,000 to 5,000 different chemicals at one time. And then we have to go through and screen them, and it's more like a cocktail when you're using the combinatorial synthesis. Parallel synthesis is making a single product at a time, so it's a lot better for targeting what we're specifically looking at. It is much more focused, but it is a much less diverse library, so we're not going to have a lot of information at one time. The procedure they use for parallel synthesis is called the tea bag procedure. So they have these porous bags, again, made of resin, because resin is not going to react, but it's going to attract the particles. And they're going to be suspended in these reagents, all depending on what it is we're trying to react. We are able to synthesize we synthesize a highly reactive intermediate via very simple series of steps. And then we're going to react these intermediates with varying different reagents. And so by creating a highly reactive intermediate, we know exactly what that intermediate is going to be. So then we take that intermediate and we are reacting it with different reagents, and that is going to allow us to pinpoint specifically which reagent is what, and it's going to be one compound versus a cocktail of compounds. This has really led to research in the structure activity of relationships because we have to know specifically what it is we are looking at. Are we looking at a receptor? So we need to make sure the drug has the design that the receptor needs. Are we looking to attach to DNA or to an enzyme, denature an enzyme? It all depends on how we're trying to target the drug. So we really need to understand the structure activity and the relationships that are going to occur between the drug and the process in which we're trying to target. So this is what we call drug optimization, is because we're making the most optimal drug for the situation. So parallel synthesis is what we want to do, but it's going to be a lot less efficient. We could do combinatorial synthesis first, identify the target cocktail, and then move into parallel synthesis as needed. The last one we're looking at is called high throughput screening, or HTS. This helps create large compound libraries because we use robotics and microscale chemistry. So this is essentially what all these processes are combined. The beauty of this is it's working with tiny little, essentially wells during this microchemistry. We can, because it's going through robotics, we're using computers to screen absolutely everything. They can look and analyze over 100,000 compounds per day while it's testing. And so we can have a large number of targets, and usually the reactions we're going to run is going to have a very measurable effect, such as a color change or displacement, has radioactive labeling, so on and so forth, to know specifically what it is we made. Moving on to what's called computer-aided design, or CAD. You've probably heard of CAD software for designing specifically houses and architecture, but that same kind of concepts goes to building 3D structures of molecules so that we can understand how they're going to interact. It's a really cool way to use technology. So this allows us to create virtual drug trials. So we can kind of go through the hiccups and the processes of drug trials, but do it all based on the computer before introducing it to any biological materials or rats, humans, whatever it happens to be. So you really kind of narrow down any kind of side effects or any kind of information you can before introducing it, so we know more about the toxic side effects, so on and so forth. We use what's called molecular modeling software, which is going to analyze the drug receptor site and the interaction that's going to occur there. So again, it all depends on where the drug is being targeted. If we are looking at a receptor on a bacterial cell, or we are looking at an enzyme that we want to denature, we need to know how the drug is interacting. So we need to know where it's going to be received. So the purpose behind this is to design drugs for the best fit possible at a receptor site. So it's much more targeted. It should have a much higher success rate. Because of our increased knowledge in biomolecular science, we know a lot more about the targets in which we're looking at. So we can use X-ray crystallography, or NMR, which stands for nuclear magnetic resonance, which will give you different readings based off the type of molecules that are in there. And that's going to help us lead to what a 3D model would look like of both the molecule and the process and the interactions that are going to occur. So what actually is created is called a pharmacophore. This is focusing in what part of the drug is the specific binding site. And so by creating a 3D version of this, we know what this site is going to look like and the interactions that may occur. So then we're going to research common features of compounds that target a receptor. We know what different receptors are and what will allow the receptor to get targeted and turned on, and so on and so forth. So by looking at common features throughout all the interactions we understand for a specific binding site, we can predict a 3D structure. So if we know caffeine is going to bind to a receptor within our brain, we know the structure of caffeine, therefore we know kind of what the structure of that receptor is going to be, therefore we can predict pretty well what the 3D molecule would need to look like, and therefore we can design a drug specifically for that receptor site. Our goal with using the CADD system is to create a database for quick searches while designing a drug. And this is going to allow us, because we know specifically what that receptor site is going to look like, it's going to reduce the number of candidate molecules that need to be synthesized and tested, because we know what it needs to look like. We know what kind of side chains it's going to need. We know if it's going to need a benzene ring, so on and so forth. All this information is going to be laid out so we can design the best drug possible. Next, we're going to look at how the structure of drugs allow for absorption and distribution within our body. So first, looking at what's called the bioavailability. Essentially, after we know how our body reacts to the drug, we can predict how much of it's actually going to make it to the bloodstream and throughout our body after digestion. Average is drug administration we shoot for is actually between 20 to 40%. Because we need to understand that we have the hydrochloric acid in our stomach, we have our saliva, there's a lot of features in our body that's going to break the drug down prior to it actually being absorbed. So we need to make sure we are designing a drug for its specific purpose so it can get to the target area and survive our digestion processes. So a couple different things we look at. Polar drugs or ionic polar drugs or ionic groups are going to increase the solubility of the drug. Because we are aqueous beings, aqueous solutions are going to dissolve polar or ionic groups that are on drugs more readily. And this is going to give you a more efficient distribution. So looking at the reaction if we have a carboxylic acid, and we want to turn that carboxylic acid into an ionic salt. So we have aspirin. And if we look at aspirin and we have the structure, we see that carboxylic acid right here with the COOH. Aspirin is not incredibly soluble. And we know that it is a weak acid because it has the carboxylic group. Because it's not very soluble, that's one of the side effects of why it's such a strong irritant to the stomach as well, because it's going to kind of sit in the stomach and not break down, therefore causing some irritants to our stomach. But to make aspirin a little bit more soluble, we are going to change the form a little bit so that when we do swallow aspirin, it's not pure aspirin. It actually has been modified so that it can be dissolved in our body a little bit easier. So we are going to react it with a strong acid, sodium hydroxide. What that is going to do is going to convert that aspirin into a sodium salt. So we have a sodium salt of the aspirin, and that makes it more soluble. So we now have that sodium replacing where that hydrogen was, and that hydrogen goes to hydroxide to make water as a byproduct. Now this is more soluble. And going back to acid bases, this is the conjugate base, and it becomes more readily broken down in our body because our body is more acidic. Therefore, being a conjugate base, it's going to react a little bit more, get broken down, and therefore get absorbed. Now we're going to look at the reaction of a basic group. So we have NH2 basic group, and we're going to form an ionic salt so that it can, again, react a little bit better within our body. So here we have the fluoxetine, which is not very soluble. And so this is how we go about actually making Prozac and what allows Prozac to become soluble. Because if we use just fluoxetine, we're not going to get the great effects of it. And so we are going to take that hydrochloric acid and we are going to react it. And so we're going to actually react it with the nitrogen itself because that is the basic group on the structure. So if we look down here, I know it got flipped and reversed, but here's that nitrogen. So that nitrogen accepted the hydrogen, and so now it's actually carrying a positive charge. And we have a chlorine ion, and so this creates this positive to negative charge creates that ionic salt due to the attractions. This is going to create a conjugate base, which is going to allow it to dissolve in different parts of the body so that it can go in specifically into the brain and get into the blood, which is more basic than our stomach and digestive system. And we had talked about enantiomers. Don't know if I'm saying that right. Looking at what do the different compounds look like. So we had touched on these back in D.9. So how do we actually produce a single enantiomer so that we know we are not going to get the two versions or like the optical isomer or so on and so forth. So we know specifically which version, which structure we are getting that is going to have the positive benefit instead of the negative consequence like we saw with thalidomide in the 60s when we realized it had an optical isomer. One dampened nausea for pregnant women, one caused fetal problems. So we use a process called asymmetric synthesis, or it's a process where we have an enantioselective process of creating a single drug with the appropriate orientation of the groups which we're targeting. So this process or the strategies we use is called chiral auxiliary. So the chiral molecule is going to bind to the reactant. It's going to block one of the reaction sites and is essentially creating what's called steric hindrance. So looking back at D8, when you can have the cis versus the trans, you can have where we have the mirror images between the two. There's a lot of different things that can happen when we are reacting different compounds together. So by attaching a chiral molecule, and so this is not going to really interact with the way the structure is, but it's going to cause the addition of a group during the reaction to go in one direction. So it's focusing on how the new molecule is going to be made. So an example is we use this with Taxol, which is a derivative from a process from the yew tree that we use for a variety of different cancers. So Taxol actually has 11 chiral carbon centers. So those are 7 or 11 centers that could essentially have different sites that we need to go through and make sure are set appropriately so that we have the appropriate effects. So we are going to use titanium or rhodium chiral catalysts, attach them to those carbon centers so that any other side chains that we're going to attach during the process of creating Taxol is appropriate. So methods using the catalysts contain the transition metals because they're a little bit less reactive and our body's not going to react against it as much.